Hello everyone, am I audible and visible? Let me know, then we'll start. Fine. I think I'm audible. Hello Kishan, good evening beta. Welcome to my class. Fine. So let us start. Hello everyone, I'm myself Dr. Deepthi Kare. I'm the PhD Physiology and your educator for Physiology. So today we'll discuss about process of muscle excitability. Okay, starting with my announcement, you will get all the soft copies of my notes. That is on the application VESALIUS. You can download the application from Play Store. Yeah, and you can subscribe the same for getting soft copies of the PDF. Second, that is if you wish to get hard copy, you can order either on Flipkart or on the Amazon. That is available. Physiology notes, that is a book which is also available. You will get all the hard copy as well as soft copy of my notes that I am teaching here. Okay, fine. Now let us start. Link for all of this that is given in the description box. Okay, welcome to my class. Now let us start today's topic, that is muscle excitability. Okay, as we all know that this is skeletal muscle. Okay, skeletal muscle, when it is excited, when impulse or action potential it passes from the nerve fiber, motor neuron, this is motor neuron. Okay, that is followed by sequences of changes, and that is followed by, this is electrical event which is followed by mechanical event, that is muscle contraction. And this combination of electrical and mechanical event, that is known as excitation contraction coupling. Early gastro GIT, the class is at 10 o'clock after completing this topic. Don't worry. Okay, 9:30 to 10 and 10 to 10:30 I'll take the class. Don't worry. Okay, fine. And here, for excitation and contraction, we require calcium ions. Okay, calcium ions are required. Okay, fine. Indian chapter, I am taking muscle physiology. You just go through book muscle excitation, contraction, coupling, and mechanism of muscle contraction. Okay, you just go through my chapter. You just first watch my class and then open the book. You will definitely come to know that which chapter is going on. Don't worry. Fine. First watch me for half an hour and then you just open the book. Okay, fine. Now, so sequences of events, as I told you, you can see here, whenever this is action potential, you can see it passes through the motor nerve. Now this action potential, it passes through neuromuscular junction, and now this action potential, it passes through the muscle, through transverse tubule. Okay, and you can see here calcium ions, they enter from the sarcoplasmic reticulum into the sarcoplasm, and this sequence is, they are responsible for muscle contraction. So this way your electrical events, they initiate contractile or mechanical events. Fine. So this is your sequences of events in excitation, contraction, coupling. Fine. Now, so as I told you, this is your axon terminal. Fine. This axon terminal where you have acetylcholine containing synaptic vesicles. Here acetylcholine is released. You can see this acetylcholine binds with the ligand gated channel. There is opening of the sodium channel. When sodium channel opens, there is depolarization, means there is action potential. This action potential has a property of propagation. Propagation means it passes. Okay, it passes along the sarcolemma. Okay, sarcolemma means the cell membrane of the skeletal muscle. Okay, then because of this, you can see here in the T tubule we have this receptors DHP. What is the full form of DHP? Dihydropyridine receptors. Fine. And this dihydropyridine receptors, they activate your other type of receptors known as ryanodine receptors. This ryanodine receptors are present here in the sarcoplasmic reticulum. This is not shown properly here, but they are present here. Okay. And when these ryanodine receptors are activated, there is opening of calcium channel, and calcium concentration increases in this fluid which is present inside the cell, intracellular fluid. Now, this calcium ion concentration increases 2000 times. What does it mean? Normal calcium ion concentration inside the cell, that is 10 raised to minus 7 moles in the resting condition per liter. Fine. Now it becomes your 2 into 10 raised to minus 4. You can see here this one. So 2000 times increases moles per liter. Fine. Okay. And now this calcium ions, they bind with the troponin C. We have discussed troponin has 3 components: troponin C, troponin I, and troponin T. We will discuss the other events now. Now, one applied aspect here is this ryanodine receptors. As I told you, these are dihydropyridine receptors. These dihydropyridine receptors are present in the T tubule. Okay. And ryanodine receptors are present here in the sarcoplasmic reticulum, in the L tubule. Fine. Now, this ryanodine receptors, if suppose some person is having genetic defect in the ryanodine receptor, what happens if we are giving general anesthesia to this patient person? General anesthesia. So what happens? There is massive large amount of release of calcium ions, and when large amount of calcium ions are released, what they cause? They cause intense muscle contraction. Okay. And that results in rapid and life threatening increase in the body temperature. This is known as malignant hyperthermia. Malignant means severe. Hyperthermia, temperature increases. Fine. Now we discuss about molecular basis of muscle contraction. So many times the question is asked. This molecular basis of muscle contraction theory was given by A.F. Huxley and H.E. Huxley in 1954. Now, the question may be asked as, it is not only asked like molecular mechanism, maybe walk along theory, or red chat theory, or sliding filament theory, or cross bridge theory, or molecular basis of muscle contraction. Now, this theory explains how this actin filaments, they slide over this myosin filament. Okay. Now we'll discuss. So first step of the cross bridge formation. So first is initiation of cross bridge cycle. So what happens in the resting state, as we have discussed? Troponin, tropomyosin, they are relaxing proteins. Means this is tropomyosin, and on which we have troponin. So this troponin, tropomyosin, they cover this active sites of actin molecule. So now this active sites of actin molecule cannot bind with myosin head. Okay. So first of all, this should be removed. How this can be removed? Because of entry of calcium ions. As we have discussed, when action potential comes, calcium ions, they come out of the sarcoplasmic reticulum into the sarcoplasmic reticulum into the sarcoplasm. So what happens? The concentration of calcium ion increases, and when this concentration of calcium ion increases, they bind with troponin C. Okay, so when they bind with troponin C, they remove this troponin, tropomyosin from actin filament molecule. Molecule of actin filament. So now this actin filament site, that gets opened up. Okay, so this is open. Now it can bind with this myosin head. Okay, so when one molecule of troponin bind with the calcium ions, 7, this is very important thing, maybe 7 binding sites on the actin filaments, they are or actin molecules, 7 actin molecules, they are uncovered. Okay, when only one calcium binds with troponin C. This troponin tropomyosin complex is removed from 7 sites. You can see here, it has got bound with 7 sites. Fine. Okay, when this lateral movement, when this troponin tropomyosin, mainly tropomyosin, this filament, it moves, this cycle of cross bridge that gets initiated. Okay, so this is first step, initiation of cross bridge cycle. Second step, that is now formation of actin myosin complex. You can see here actin binds with the myosin. You can see here, now this active site of the actin gets free to bind with the myosin. Okay, so you can see here, first of all, before that, your myosin head requires energy. So for that, what happens, your myosin head gets energy from the ATP. It binds with the ATP. Then myosin head has got enzyme ATPase, which degrades ATP into ADP and phosphate. Okay, now it releases ADP and phosphate gradually, and because of this release, there is and the energy produced because of breakdown of ATP, what happens, now this myosin head that gets attached to the actin molecule, and there is also change in the shape, conformational change, 90% conformational change occur, and it causes your power stroke. It gives power stroke. I'll show you. So this is your power stroke, you can see here. So in power stroke, what happens, formation of, you can see here, actin, this is active site of actin, myosin, ADP and phosphate. So first of all, what happens, there is release of ADP and phosphate. Okay, and followed by that, whatever energy is released, that causes conformational change in the myosin head. So you can say that myosin head gets the power stroke, means it becomes slightly flexed. Okay, so flexion of the myosin head towards the arm part. Sorry, here you can see, this is the head part and this is the arm part, so it gets flexed. Okay, and this, the new alignment of the forces, they cause head to tilt towards the arm, and along with, when the head moves, along with it, it also moves your actin filaments medially. Okay, so tilt of the head, that is your power stroke. Fine. Now, so power stroke has two effects. Number one, if the load is small, means small amount of actin filaments are there, we are not lifting much load. So what happens? There is sliding of the actin filaments. Okay. But if the load is large, what happens? That actin filaments, they are unable to slide. Okay. So you can say that myosin head get flexed. Okay, more, and it producing more stretching of the myosin head, because here the force is large. Fine. Now next step is, now what happens, there is detachment of the head of a cross bridge from the active site of the filaments. Okay, so what happens here, after your ADP and phosphates are released and there is power stroke, what happens, now your next ATP binds, and at that time, when your next ATP binds, what happens, your myosin head gets detached from the previously bound actin molecule. Okay, so myosin head detached from the actin molecule of the actin filament. Okay, and after that it gets reactivated. You can see here, these are the steps. First, myosin head attached to the actin. Okay, then there is release of phosphate and ADP that gives energy to the myosin head, and myosin head gets flexed, and flexion of the myosin head causes movement of the actin filament. Okay, then next step is, when new ATP binds, this head detached from this previously bound actin molecule, now it binds with the other actin molecule and it causes movement of that actin molecule. So continuously, like we are moving the pages of our book, this way you can see here, suppose this is the page, so it is binding with this, then it causes like this, then it binds with second, then it causes like this way. So your actin filament moves medially. Okay, so this is the basic mechanism. Let us repeat with the help of diagram. You can see first step, you can see here ATP binds. Okay, cycle begins with the binding of ATP to the myosin head. When ATP binds with the myosin head, what happens? This head gets detached from the previously bound actin molecule. Fine. Then next step is now ATP is degraded because the enzyme is ATPase. So ATP gets degraded into ADP and phosphate. Okay, fine. So now what happens? ADP and phosphate are formed. Okay. Then third is now there is gradually release of ADP and phosphate, and because of hydrolysis and release of ADP and phosphate, energy is released, and this energy leads to there is power stroke. Okay, or you can say conformational change in the head. Okay, so you can see here there is fourth stage here, release of ADP and phosphate and ADP here, that gives you power stroke, and because of power stroke that actin filament moves medially. Okay, then last step again, new ATP molecule binds, and when new ATP molecule binds, then what happens? It releases myosin head from the previously bound actin molecule. Now it binds with the next molecule. Fine. So this way, this is the actual cycle. You can see first step here. This is myosin head bind with number 1 molecule. You can easily remember here. Okay. Then what happens? ATP binds. When ATP binds, this is detached from number 1. ATP is degraded into ADP and phosphate. Okay. Now what happens? Because of release of energy, because of release of ADP and phosphate, now this head gets energized and it binds with number 2 molecule. You can see here. Okay. And there is flexion. So it moves this number 2 medially, this side. Again next ATP binds, then this gets detached from number 2 and binds with number 3. Okay, so this way gradually it binds with one by one actin molecules. Fine. Now next is steps in the muscle relaxation. What happens? Very important thing here is muscle relaxation is also an active process. Why active process? Because it requires ATP. So after few milliseconds, when action potential is over, what happens now? Your calcium ion concentration in the sarcoplasm is reduced. How? Because this calcium ions, they go back into the sarcoplasmic reticulum. And this requires your calcium pump, because this is also an active transport and it requires energy. Fine. Next is here the same thing is there. Skeletal muscle has both contraction and relaxation as active process. Okay, calcium ions, they go back into the sarcoplasmic reticulum, that is also an active process. Fine. Now very important thing here is why there is rigidity after death. This rigidity after death is known as rigor mortis. Okay, rigidity after death. Okay, what is the cause of this rigidity? Because after death, as there is no oxygen, okay, because person is not taking breath, so what happens? There is aerobic glycolysis is absent. Okay, so what happens? Aerobic glycolysis means here glucose is aerobically aerobic oxidation of glucose, and that produces 38 ATPs. Okay, Krebs cycle and all, you all must be knowing. So here no aerobic glycolysis, no ATPs are generated, and because of lack of ATP, you can see here ATPs are not there. So what happens? Now there is no back entry of calcium ions into the sarcoplasmic reticulum. So here what happens? There is a fixation of cross bridge here. Cross bridges are formed and they just cannot be detached. No detachment of the cross bridges. Okay, so there is no relaxation. And this is, you can see here as rigor mortis. Now what is rigor mortis? I define it. Rigor mortis, it is a state of shortening or rigidity that presents in body muscle rigidity, after death. What is the cause? Because of lack of ATP. Fine. So muscle cannot relax and muscle are in a state of rigidity, that is rigor mortis. Okay, so myosin heads, they get attached to the actin molecule. Fine. And after death, you can say that calcium ions, they will not go back into the sarcoplasmic reticulum. Now, what is the importance of this rigor mortis? Now, first of all, mechanism, as we have already discussed, because of increase in the calcium ion concentration, okay, lack of ATP and calcium ions, they will not go back into the sarcoplasmic reticulum. So there is rigidity in the muscles. Okay, fine. Now, what is the importance, medico-legal importance of the rigor mortis? You can see here, this rigor mortis, it starts appearing after, you can see, around 3 to 4 hours after death. Okay, 3 to 4 hours after death, and it gets completed by 12 hours. Okay, and the stiffness disappears by maximally by 48 to 60 hours after death. Here you can say that rigor mortis will not disappear, but there is destruction, disintegration of muscle proteins. Muscle proteins are destroyed, disintegration of muscle proteins. So you will not find exit rigor mortis. Otherwise, it is irreversible process. Okay. So rigor mortis has medico-legal importance. We can come to know the time of death. Time of death. If we know the rigor, means suppose if rigor has not appeared, then you can say that the death occurred 3 to 4 hours before. Okay, if rigor is completely developed, you can say that there are 12 hours. 12 hours have completed. Okay, fine. So this is all about rigor mortis. Last but important thing here is what happens when there is cross bridge cycle formation. Okay, you can see here, this is what are the changes they occur in the sarcomere changes at when there is cross bridge cycle. You can see here, this is the relaxed state, this one. Okay, and this is the contracted state. What happens here, A band will not change. You can see here, this A band remains same. There is no change in the A band, very important. Then H zone disappears. This is your H zone. When muscle contracts, H zone means it is the zone where only myosin filaments are there, not actin. But what happens when muscle contracts, actin filaments are also overlapping, so your H zone disappears. Okay, I band also decreases because I band consists of only actin filaments, this one. And now actin filaments are moving medially, so I band also decreases. Z line, this is Z line, you can see here. Z line also comes, they also come close together. Sarcomere also gets shortened. Okay, and actin filaments from the opposite side, opposite end of the sarcomere, they approach each other. Okay, so these are all the changes they occur during cross bridge cycle. Okay, so this is all about our today's topic, that is muscle excitability and you can say molecular basis of muscle contraction. Okay, fine. Thank you. One announcement again, that if you wish to get all my notes, they are available in soft copy and you can find it on the application VESALIUS. For that you have to download the application VESALIUS from Play Store. Okay, and subscribe for the same. Link for that is given in the description box. Or if you wish to get hard copy of my notes, you just have to go to Flipkart or Amazon, and link for the same is also given in the description box and you can order the same. Okay, thank you so much. If you like my video, you can like it, you can share with all your friends, and you can subscribe my channel. And all, please hit the bell icon. Why? Because if you hit the bell icon, then you can come to know that madam is online. Okay, lecture has been started, so this is very important. Just hit the bell icon. Okay, thank you so much.